Hello and welcome to our next unit. In this unit, we are going to start talking about cells, which are the smallest thing that can actually be considered alive. So in our previous unit, we were talking about molecules. We talked about proteins and lipids and carbohydrates. Individually, these things are not living. However, when they come together in certain arrangements, they can be making up a cell. A cell is the smallest thing that can carry on all the functions of life, and so cells are the smallest thing that is considered to be alive. So we haven't known about cells for an incredibly long period of time. If you consider the fact that humans have been around for an extremely long time, we first discovered cells in the 1600s. So we haven't known a ton about them, and what we're learning about them increases every single day. So in 1665, there was an English guy named Robert Hooke. He actually took slices of cork and sliced it really, really thin and looked at it underneath the microscope. He gave what he saw were little boxes, and they reminded him of the bedroom of a monk. A monk's bedroom was termed the monk's cell, and so he gave these little boxes that he saw the term cell. For those of you that don't know, cork is actually dried pieces of tree. And so what he was looking at was plant cells underneath the microscope. He was just the first one to actually give it a term and to start to describe them. In 1673, a guy named Anton von Leeuwenhoek, he was the first to look at living cells. And so what he did was he actually got samples of pond water and looked at those underneath the microscope. And underneath the microscope, he saw all these little tiny things moving around, and started writing about them and recording the information that he saw about them. So then we go forward a couple hundred years. We have a guy named Matthias Schleiden. Schleiden looked at a whole bunch of different plants and different parts of different plants and made the very profound and important statement that all plants are made of cells. I remember that Schleiden was the one that talked about plants because of the L in Schleiden and the L in plants. In 1839, a year later, another guy who was working similarly to Schleiden. So Schleiden was looking at all different parts of plants and all types of plants. This guy, Theodor Schwann, he looked at all different types of animals and different parts of animals, and he came up with all animals are made of cells. I remember Schwann is animals because Schwann sounds like swan, and so a swan is an animal, and that's how I remember that Schwann was animals. 1855, a Polish guy named Rudolf Virchow comes up with a very profound and important statement that cells come only from other cells. This is huge because up until this time, they believed in something called spontaneous generation. And so spontaneous generation said that living things came from non-living things. So for instance, mice came from a pile of rags, or mud would give you frogs. And so for a very, very long time, they believed that spirits and other things like that would give rise to organisms. So for Virchow to come along and say, no, cells come from other cells, and therefore organs and organisms come from other organisms, was huge because, again, it went against what the common person believed. Collectively, these five people, their work comes together to make what is called the cell theory. And the cell theory says that all living things are made of cells. Cells are the basic unit of structure and function, and cells come from the reproduction of other cells. This is important. This theory is something you need to know. To us, this all seems like common sense. Well, no kidding, cells come from other cells. Yeah, we know every living thing is made up of cells. But you got to remember that in the 16 to 1800s, when they were saying these things, this was huge and new and profound information. To us, again, it seems really obvious, but at the time, these were profound statements. So this collective theory is the basis for what we now know as how organisms function. There are two major features related to cells that you need to understand, and the first is that cells are limited by their size. Most cells are between 10 and 50 micrometers in diameter. So remember, 1,000 micrometers is equal to 1 millimeter. Okay? So most cells are really, really tiny. You can't see them without a microscope. What's limiting them is the ratio between their outside surface area and their inside volume. Volume is going to increase more rapidly than surface area because surface area is squared, volume is cubed. And so what this means is it becomes important for the cell to be able to exchange nutrients, to get nutrients into the cell that it needs, to get gases in that it needs and get rid of gases that it doesn't want, to get waste products across that cell membrane. The bigger the cell is, the harder it is to do that. And so size is going to limit the surface area to volume ratio is going to limit the size of the cell. The other important feature for cells is shape. Shape directly controls the function. How the cell works is directly determined by how the cell is put together. And this is a theme that we're going to see throughout the rest of the year. And we've already seen it, that how the molecule works directly determines how it functions. That saturated fats having no double bonds means that they can pack tighter, which means they're harder to break down. Whereas an unsaturated fat having the double bonds means that it can't pack as tightly, so it's going to be a liquid. That shape directly relates to its function. And so this is a theme that's going to carry through into cells as well. Inside the cells, there's what are called organelles, which literally means little organs. These are internal structures that do the functions of the cell. Couple of them include the cell membrane. All cells will have a cell membrane, which is going to surround the cell and be a boundary. It's going to protect the cell and allow some things in and keep other things out. And then what we call prokaryotic cells do not have a nucleus, but eukaryotic cells do. Eukaryotic cells are going to have genetic information in them, specifically DNA, located inside of a nucleus. And so the DNA is going to be inside your nucleus, which is then inside your cell. So these really important directions that are going to direct everything for the cell are located in the center in a protected environment. Okay, so eukaryotes versus prokaryotes. The term karyote refers to a cell. The prefix "eu" means true. Okay, so a eukaryote is what's referred to as a true cell. Eukaryotes have organelles that have membranes around them, and they have a nucleus that has a membrane surrounding it. The prefix pro, means before. Okay. So prokaryotes came before eukaryotes. So prokaryotes, they don't have a nucleus in them, and they don't have any organelles that have cell membranes around them. All prokaryotes are unicellular, but not all unicellular organisms are prokaryotes. You can have eukaryotes that are unicellular. One thing that is pretty much synonymous is that all bacteria are prokaryotes, and prokaryotes are all bacteria. Okay? So prokaryotes and bacteria, they're the same thing. It's just a different term for it. It's talking about the types of cells that are involved. Okay? So prokaryotes are very simple. They don't have a nucleus in them, and they don't have a ton of different organelles. Whereas eukaryotes are much more complex. They do have a nucleus, and they have tons of different organelles. And we're going to talk about all of those organelles in a lot more detail in class. As always, if you have any questions, please write them down and ask in class.